Okay, next up is Dr. Robert Dirksen and Philip Van Paggen. Um, both of these people are on the Scientific Advisory Board. Dr. Robert Dirksen is a professor at the University of Rochester and chair of the RYR1 Foundation Scientific Advisory Board. His research focuses on how RYR1 variants affect skeletal muscle function using mouse models to better understand the disease and explore potential treatments. We're also fortunate to have Dr. Philip Van Paggen, a professor at the University of British Columbia. His work focuses on the structure and function of ion channels, including the RYR1 protein, revealing how mutations and molecular interactions impact its behavior. Together, they'll be presenting mechanism and functional readouts of RYR1 variants, a session that will help us understand using clear examples how changes in the RYR1 gene cause muscle problems and how researchers study those changes in the lab to better understand the disease and guide treatment development. It's a privilege to have both of these leading experts with us today to share their knowledge and insights with the RYR1 RD community.
Thank you very much. It's really a delight to be back here in Pittsburgh at the Family Conference. This is my favorite meeting I ever go to. And as was said last night, it's not really a family conference, it's a family reunion. It's great to come back and see family members and see how things are going with them and to meet new people, just as I would at my own reunion. So it's really great to be here and have an opportunity in this lecture to tag team with Philip. We're going to kind of be the Hulk Hogan and Rock of this meeting, where he looks more the part of Hulk Hogan than I do, but I'm going to do my best to be the Rock. I'm going to talk a little bit in this about the mechanisms, and then Philip will move on to the structure and how we can understand more about mechanisms from the structure. And I'm a muscle physiologist. I like to understand how muscle works. And much like you would take your car to a mechanic who would put it up on the lift and look through all the different parts of the car to figure out what's wrong with it, and then diagnose that problem and then replace the part and give you back a working car. I'm a muscle mechanic. I want to understand how every part of the muscle works, including the RYR1 receptor and how it works with all of its partners. And in RYR1 related disease, we want to understand what's going wrong. And if we can understand that, then we can develop therapies that target those issues. So that's what I'm going to try to address here in this lecture. At a very simplistic way, muscle force is generated by a calcium signal, right? So normally in the resting muscle, calcium is very, very low. And there's a range which calcium can increase and it doesn't cause any contraction. But then during muscle contraction, when you want to generate force, move your bones, you need to elevate calcium, and that's the job of the RYR1 receptor. And there's a fairly linear relationship, or sigmoidal relationship, where the more calcium gets increased, the more the contraction, the stronger the force, the more we're able to generate force and our strength is higher. And in RYR1 disease, the RYR1 receptor is not fully functional, and you produce less force, and you have weakness. And so the lack of the RYR1 receptor in the disease state to produce a full calcium signal results in some weakness. And we try to understand how variants in this RYR1 receptor result in this reduction in calcium release and force. So we know the RYR1 receptor is really important. It's actually absolutely critical for life, because when we remove the RYR1 receptor from mice, they die at they're embryonic lethal, or they die at birth because their muscles don't work and they cannot breathe at all. So the RYR1 receptor is absolutely required for life, and we want to understand then, why is it so important? Karsten did a great talk already about the excitation, contraction, coupling machinery, but I just want to kind of revisit that for a second. We have our nerve, where our central nervous system says we want our muscle to contract, so it sends a signal to the muscle cell to contract, and the way it does that is it creates an electrical signal in the surface membrane of the muscle cell, which we call an action potential. It moves very, very quickly down into that specialized structure that he mentions, the triad, which is where all the activity of the RYR1 receptor happens. And as you can see in this cartoon, the RYR1 receptor is controlled by this other protein called the dihydropyridine receptor that's in the triad too. And so they're kind of connected by this coil or this linkage of a chain. And when an action potential comes down, the T-tubule, or into the triad, it activates that dihydropyridine receptor to open the RYR1 receptor and to release the calcium. So in a relaxed muscle, when there's no action potential, these RYR1 receptors are closed, the calcium is in the store. The myofilaments here that need calcium to contract are relaxed because there's very low calcium in the muscle, and so we have a relaxed cell. But now when we excite this cell, the nerve initiates an electrical signal that triggers that dihydropyridine receptor to pull on that chain and open up the ryanodine receptor to release the calcium that then sprinkles down on top of our myofilaments. And under this condition, the RYR1 receptor channels are open, calcium is released, calcium increases in the cell, and as we showed in that last graph, the more the calcium increases, the stronger the contraction, and we get our muscle contraction. So if you've been at any of my lectures in the past, I've always made the analogy that this whole process is very similar to a toilet. And if you think about the toilet as the reservoir for calcium, and the water is the calcium itself, so you've got this reservoir of SR full of calcium, the connection in the triad between that dihydropyridine receptor and the ryanodine receptor is analogous to the handle on the toilet that is connected to that chain that's linked across the triad junction to the ryanodine receptor, which is the flapper, right? So the flapper here stays closed, but when there's an action potential, it tugs on the dihydropyridine receptor, it pulls the chain, it opens the flapper, and then calcium can rush into the toilet bowl here, which is the cytoplasm, and that's where all the good stuff is, the myofilaments that cause muscle contraction, okay? So this is a very simplistic way of kind of looking at how the EC coupling machine works in an everyday situation. So in the disease, Carson also talked about the different forms of the disease and the genetics. We know that RYR1-related disease has both dominant versions and recessive versions. The dominant versions include malignant hyperthermia, central core disease, some forms of multi-mini core disease, and recessive, which are more loss of function, results in multi-mini core disease, some forms of central core disease, central nuclear myopathy, and congenital fiber disproportion. And in general, the dominant disorders are a gain of function. The RYR1 receptor has an increased function or a change in its function, and the recessive is kind of a loss of function. And generally, you have a more severe and earlier onset phenotype with the recessive forms. So then how do these mutations result in altered RYR1 channel function and calcium release and contraction? And so we can see this through the central dogma of DNA to RNA to protein that Olivia went through. We have our exons that are spliced out the introns to make the normal RNA. The RYR1 receptor is a tetramer of 4 identical proteins that come together to make a channel. If there is a mutation in one of those exons, then you're going to make a channel that has a variant in it, and each of those variants can come together to make the tetrameric ion channel. And in dominant inheritance, remember, we get one copy of each of the gene or the allele from each parent, and we have in the dominant, we have a normal allele and one with a variant, and so then you produce subunits or these little pieces of the channel that are either control blue wild type or normal, or then one with the variant. And so then you get different combinations of 1, 2, 3, or 4 variants. And obviously, some of the variable expressivity might have to do with what proportion of mutant or variant subunits there are to wild type, and that may even differ from one muscle to the next. In recessive, we have variants in both alleles. They could be identical variants, for example, that create identically altered proteins. Or you could have a stop codon, a nonsense codon, that doesn't create a transcript that's stable and doesn't create a protein. And so you're only producing much less protein that, again, has a variant in it. So in this, you can understand why this would be more severe than these conditions, because you have less protein, and the protein you have has a variant in it. It's not normal. So over many years, we've kind of looked at the different mechanisms by which this communication between the dihydropyridine receptor and the RYR1 receptor might occur. This is shown schematically on one side of the triad, where we have the surface membrane or T-tubule up here with the dihydropyridine receptor connected with that chain to the opening the gate of the ryanodine receptor here, working normally. In some of the dominant gain of functions, the ryanodine receptor is leaky, and it produces some leak of calcium out of the SR, which is going into the cytoplasm. Maybe is not high enough to cause a contracture, but high enough to start to cause damage over long periods of time to the muscle, and also depletes or reduces some of the calcium in the store, and therefore you're not going to have as much calcium to release to cause contraction. Other forms, so that's right. So I actually put together a short video of what a leaky channel might look like, because Philip's going to show really good high resolution structures, and I wanted to give you my high resolution structure of the toilet with a leaky ryanodine receptor. This is a very short video.
I've been having problems with my toilet. Check it out. If we look into the toilet bowl, you can see that the water is rippling. That means that there's water leaking in from the tank into the bowl. Just about every time a leak like this happens, it is because of this flapper right over here. Water is leaking by. It's not being sealed off by the flapper all the way.
And so that's basically creating this leak into the cytoplasm that's putting extra calcium in there, that's causing more damage, and also reducing the amount of water in our tank or the amount of calcium in our SR needed for the next contraction. All right, so the other mechanism we found is there are some mutations in the pore region of the channel that make the channel not release calcium very well, and that obviously is going to be bad, because if calcium's not going to be released well, it won't work well. And then with the recessive variants, you obviously have the combination of maybe a leaky variant in the presence of very low channels. So this is going to be a very severe condition, because we don't have many channels that are working, and the ones that are there are not working normally. So you're going to have kind of a combination of leak and a loss of the ability to release calcium. So these are some of the underlying mechanisms. I'm going to turn it over now to Philip, who's going to continue with maybe more on the structural side of how these variants alter RYR1 receptor.
Okay, well, thank you, Bob. And I want to tell you a little bit more about actually the structure of RYR1 and what this can tell us about how disease mutations or sequence variants can actually alter it. And so, you know, in essence, right, the RYR1 is a channel, as we've seen. And what it wants to do is to release calcium, right, from this compartment that's called the sarcoplasmic reticulum into the cytosol. And so it's a channel, which means it actually has the ability also to open and close. And this is actually critical. It's not just something that's sitting there static. It's a channel that can open and close. And what we'd like to understand is, how can it open and close? And how do disease-causing mutations affect that? Now the problem is, right, the ranitidine receptor, it's a channel, it's a protein. How big is that? Well, it actually is around 0.000001 inch in size. So the question is, how can you visualize that? You know, we kind of need X-ray vision to see that. I don't have X-ray vision. This guy has X-ray vision, but I don't. So this is not something you can see with the naked eye. You can think we've developed tools, right, to magnify things. A magnifying glass, unfortunately, isn't going to do it. Of course, you think, well, what about microscopes? Surely with a microscope, you should be able to see that. But in fact, a microscope, what we call a light microscope, still isn't powerful enough to really visualize RYR1. What you actually need is a very fancy instrument called an electron microscope. And so here's an example of an electron microscope. It's a fancy machine, costs about $10 million. It's extremely powerful, and what we do is we shoot electrons at proteins like RYR1. And this actually allows us to start to see something. I'm going to show you an example of an actual image that you can see of RYR1. You're looking here at a very grainy image, and you can see here, this is actually purified RYR1. And if you squint your eyes, you kind of see these little darker regions. Those are actual individual RYR1 proteins that we can see directly with an electron microscope. So it's extremely powerful, and in a way, you could say, well, this is maybe impressive, right? Now we can see a protein directly with an electron microscope. But if we're being honest, look at each one of these little blobs, kind of blobby. It's a bit of a crappy image, right? How can we understand, this complex function when all we get to see is like little dark blobs? Well, here's a little trick we can do. What if we take these images and we take hundreds of thousands of images, and each time we get a bad image, but then we superpose them onto one another. All right? So if we're sitting in the dark and I take a picture of your face with a very brief exposure, I'm not going to get a good image. But if I can take that image 1,000 times, right, and we superpose them onto one another, we can actually enhance the contrast. And so this is what we do here. We're going to image this thousands of times, and you superpose them, and now you start to see some interesting features. Now you can actually start to see some details of this RYR1 protein. Now what you can now do here is actually a lot of computational steps are involved, but when you have a lot of images of a top view and a side view and a tilted view of the same object, we can actually create a three-dimensional structure. If I was to take an image of you from several different angles, I can reconstruct you in 3 dimensions. And so this is exactly the same thing we can do with RYR1. And so this is how RYR1 looks like. So this is the direct result of electron microscopy imaging of RYR1. All right, looks like a very complicated object. Dr. Dirksen calls this a toilet. I don't know what kind of toilet he has at home, but this does not look very comfortable, right? I rather call it like a sort of mushroom, right? And here we're looking at the top of the mushroom. Very complicated. But how complicated is it exactly? Well, let's do a little quiz here. Does anybody know this molecule? All right, small molecule here, water. An oxygen, 2 hydrogen atoms, H2O, water, right? How big is the ranitidine receptor, RYR1, compared to this? Let's have a look. Water, we need to zoom out. Every sphere you see here is an atom. Okay, so this is how big RYR1 is relative to a water molecule. All right, a water molecule has 3 atoms. RYR1 has 315,832 atoms. Okay, it's incredibly complex. And I've been staring at this for many years. We've been working on this, and others in the field as well. I'm not going to claim that I can, completely keep these 315,000 atoms in my head. I don't think anybody can. But there's a lot of interesting things we can do with this, because we can visualize it, and we have computational tools to start analyzing this. Okay, one of the things you can ask, how does calcium actually move through this channel? And we're looking here again at the view of the ryanodine receptor, and it's actually rotate a little bit. We're looking here at the top, and what we're going to go now is go inside the protein, and we're going to take a look. And actually, if you take a look here, we're going here deep into the channel. This is the part actually where normally calcium ions would cross into the cytoplasm of the cell. And you can see here, this is actually blocked off, because what I'm showing here is a three-dimensional structure of RYR1 that's in a closed state. It's in a way that calcium ions cannot pass. So this is normally when your muscles are resting, RYR1 should be closed. This is what it's supposed to look like. So how does it open? I'm going to show an animation that shows you what happens when the ryanodine receptor opens. If you look carefully, it might be a little hard to see. You see deep down there, that is actually the channel part of RYR1 that's opening and closing. And it's actually a pretty small movement, but it's big enough that when it's open, calciums can come through and will drive your muscle contraction. But I hope what you can see here, there's a lot of additional movements happening, right? You see a lot of things, lots of bits and parts of this RYR1 is moving as we open the channel. And actually, if you look carefully, I want to focus on this. You see here two little blobs, maybe hard to see from the back. If you look carefully, there's two parts of the ryanodine receptor that, when the channel is closed, they're touching one another. When the channel is opening, it's pulling apart. And it's actually very important to understand disease mutations. So these are parts that actually, they like to interact with one another. They have attractive forces. And if you want to open the channel, we actually need to pull that apart. And if you didn't see that, let's zoom in a little bit, right? So we have here these two parts of the protein. As the channel is opening, these two parts of the protein, they need to be pulled apart. And it's actually extremely important, because what this part of the protein is doing is actually preventing the channel from opening too easily. Because we actually know that these two parts of the protein, they really like one another. There are attractive forces between them that prevent RYR1 from opening too easily. So think about it as follows. Right? Here's a schematic view of RYR1. Here's the pore of the channel, it's closed, right? And there are these attractive forces. We actually know there's negative charges and positive charges, and they really like one another. They prevent the channel from opening. If you want to open the channel, you need to pull that apart. And that actually is going to cost some energy. There are actually other things that need to happen before RYR1 will open. This actually prevents it from opening too easily. The way to maybe visualize it or think about this, think about if you had little magnets here, right? You want to pull these things apart. These magnets are going to keep that thing shut, right? So normally, under normal conditions, if you want to open RYR1, by default, it wants to stay closed. It needs a trigger. It needs some kind of stimulus to open it, because there's all these parts of it that want to keep it closed. Now, what does that mean for sequence variations, for disease mutants? I'll just give you one example. So, for example, you've seen like genetic reports, right? And it's like, okay, you get this complicated looking thing. Maybe you have a genetic report and it says, okay, you've got sequence variants in RYR1, ASP166 glycine, D166G. That's an amino acid number change. Now, by itself, what does that mean? It's a little hard to interpret, right? You have all these things. Surely something has changed in RYR1, but what has actually changed? Now, the nice thing with these three-dimensional structures is that we can directly map the location of this variant on the three-dimensional structure. And I'll show this here. This actually this little part here, shown in black, that is actually the part that is affected by that particular sequence variant. And in fact, that particular sequence variant affects these two parts of the protein that attract one another. So you can imagine what's going to happen here. We're actually affecting the part that wants to attract one another, that's keeping the channel shut. And so we're now putting a sequence variant in there. It's going to change the protein a little bit. They're not going to attract one another very well anymore. And as a result, it's now going to be easier to open the channel. So think about it as follows, right? You have your little magnets here. The magnet is broken. The attractive forces have been reduced. And what does that mean? That actually means it's going to be a lot easier to open RYR1. And unfortunately, that's just then what happens in disease, that because these attractive forces are disrupted, the channel will occasionally open by itself. And when it opens by itself, calcium will start to leak through, and that, of course, we know can cause muscle damage and muscle fatigue. Now, this is one example of one very specific sequence variant. We know there are over 700 different sequence variants that have been associated with disease. And this is by no means all of them, but there's so many, and every single sequence variant pretty much has its own story, its own mechanism by which it can open or close RYR1. But the nice thing is with three-dimensional structures, right, we can always go in and really look in a fine detail at the very atomic level, try to understand how does each sequence variant work, and what might it actually do, and how could it change the function of RYR1. Now, another thing you can do with this method is think about drugs. Now, I had a quiz about H2O, which is kind of an easy quiz. This is going to be a hard one, but let's see if anybody knows this molecule. Dantrolene. Oh, yeah, very good. This is indeed dantrolene, which maybe some of you are taking. Okay, I'm impressed. Okay, dantrolene. We know that dantrolene, right, is fantastic during malignant hyperthermia episodes, right? It's a life-saving drug. So you can ask yourself the question, well, how does dantrolene work? Where does it bind on RYR1? Here is a structure of RYR1. I'm going to see who can tell me where dantrolene binds, because it's actually shown. Dantrolene is bound here. It's small black sticks. It's like looking for a needle in a haystack, right? You've got this monstrous protein. You've got the small dantrolene molecule. Don't know if you can see it yet. Zoom in a little bit. There you go. A tiny molecule of dantrolene binding to the giant ranitidine receptor. Right? It's like David versus Goliath. This small molecule binding here to this tiny little pocket of RYR1 is able to keep RYR1 shut down. And actually, for completeness, we actually know that the RYCals, right, actually they bind to the exact same location. So actually the mechanism by which dantrolene and RYCals can work to prevent calcium leak is probably very similar. Right? And so this kind of information can give us clues not only on how does the drug actually work, but it can also give information about how might we modify this drug to actually make it fit better, maybe become more potent, a more potent inhibitor, and that could have very good therapeutic consequences. Okay, so with that, I just want to thank you for listening. There have been various people in my group, and also people in other labs who have been working on ranitidine receptor structures. And thank you all. Yeah, so disclaimer, I don't see patients, right? I'm a basic scientist. So yeah, the short answer is no, I don't see them. Now, dantrolene, I know, and you can probably talk to other people. Sheila Riazzi is here. I know there's patients that are receiving and are actually taking low-dose dantrolene, kind of as a regular supplement. The only thing I can tell you is that with dantrolene, you have to be careful for liver toxicity. So you have to stick to low dose. I think, without giving you too much medical advice, is something to be careful with. But anecdotally, many people have suggested that it alleviates symptoms. But in the long term, if you want a high dose of dantrolene, that's okay during malignant hyperthermia, because it's a one-time shot. You don't want to take a high dose of dantrolene every day, because your liver will suffer. And that's why it's important that there are other drugs in the pipeline that can do the same thing without having liver toxicity.
Can you use the microphone?
Thank you. Are there any trials of small molecules or ASOs to inhibit ryanodioid to fix the calcium imbalances?
Yeah, so the one trial, there was a trial that was actually started together with RYR1 Foundation, is a so-called RYR1 trial that many people know, also known as ARM210, which now has been renamed to something different. So the phase 1 clinical trial was finished. Phase 2 is being set up, and I don't know if the people involved with the trial are actually still here. I don't know, Mike, maybe you can comment to that.
I have a whole session this afternoon on different drugs that are currently available, like dantrolene. RYR1's not technically available, but we're going to address a lot of these issues this afternoon. We have a whole session on therapies, either currently available therapies as well as things that are kind of perhaps several years down the road.
I can tell you that there are many other molecules in the pipeline, right? So dantrolene is the one that's available, has some issues. RYR1 is the next thing that's already moving in clinical trials, but in many academic labs, many labs are working on new molecules that can also inhibit RYR1, and that will hopefully hit the pipeline and move into clinical trials. And the more drugs we have, the better, right?
Thank you.
So I just have one question for Dr. Van Peteghem, but I just want to sort of preface it. Are people familiar with some of the therapies for Duchenne's where they're using a truncated protein of dystrophin? Is anyone familiar with that? Okay. Well, I will tell you that for Duchenne's, one of the currently available gene therapies is they're using a gene that encodes for a smaller version of the dystrophin protein. So the analogous situation for RYR1, in theory, could be, why not have a smaller gene that would encode for a smaller or truncated ryanodine receptor protein? I'm wondering, Philip, given what you just did, is so elegantly describing the structure and function of the protein, can you explain why that probably isn't possible for RYR1?
Yeah, so RYR1, it's a big protein. And there's a reason it's so big, is that it needs to be regulated by many molecules, different proteins. RYR1 is actually expressed not only in the muscle and other tissues as well. And what we know about the structure is that pretty much every element is essential. As soon as you take one block away, either the whole house kind of falls apart, it can't do its job anymore, or it actually starts to become very leaky. And we know that the RYR1, it forms a channel. It needs to open and close. And you can actually minimize that. We actually know that you can take a small portion of RYR1, and it can form a channel perfectly on its own. But it will do so in a very dysregulated fashion. And particularly what Dr. Dirksen has talked about, the DHPR, right, that controls the flap of the toilet, that actually involves a part of the RYR that's pretty extensive. And so as soon as we were to take any of those parts away, we're going to miss out on the essential regulation of the protein. And there are a couple of regions that we can, and I know if we've conversed about this before, a couple of regions that we can probably cut out, but the bulk of the protein, I'm afraid it's not something that we can touch and start to cut out.
So, yeah, perfect. So, I mean, this comes up, this is going to be very interesting, but just real quick, Hitchum, this comes up all the time when I talk to biotechs who I'm trying to convince to study RYR1 or develop a treatment, one of the first things they will ask me is they'll say, well, in Duchenne's, they're coming up with a smaller version of the protein that isn't perfect, but it's like good enough. Why can't you do that for the ryanodine receptor? And yes, I recently was conversing with Philip about this issue, and just the complexity of the protein, the fact that it's 4 different subunits, and how it interacts with each other, as well as these other proteins that are external to it, it just doesn't seem feasible, and therefore we're stuck dealing with the entire protein. Haichum?
Great talk, thank you very much. So I was wondering now, with all these data that you generated with years, can we model other channels that are small enough to be delivered? Because RYR1 is not the only calcium channel. And now with artificial intelligence, do you think that one day we can model a smaller channel that can replace ryanodine receptor, like interacting with the DHRP or PR and replace RYR1 channel?
Yeah, so it's kind of on the same theme, right? Can we make something smaller, whether even just truncating or something from scratch? I predict it's going to be very hard. Not that AI can't make a new channel, because that's definitely possible. But think about it. So RYR1 in a native muscle context, right? I'm showing a structure of an individual RYR1, but we know it makes interactions with neighboring RYR1s. And that requires this massive, cap of that mushroom is forming all these additional interactions. So that's one part that actually probably needs to stay there. Then you have the part that interacts with the DHPR. That needs to stay there. We have the ligands that can modulate it, ATP, calcium. They probably need to stay there because they provide additional input to the channel. So putting it all together, and if I'm thinking, what could you take away? It's really hard, because you could create a channel that perhaps could interact with DHPR, but then you're not going to recapitulate the interactions between RYRs and all the additional proteins involved. So that's why I don't really foresee that's going to be possible. Yeah.
Carson?
Yeah, I want to follow up on the Duchenne analogy. And so in DMD, there is another gene called utrophin that is structurally very similar to the dystrophin gene. And one of the therapeutic strategies is to upregulate this related gene in muscle to make up for the missing dystrophin gene. So in RYR land, there is RYR3, for instance. So is that an option to even think about if we were to find a small molecule or genetic strategy to upregulate RYR3 in muscle to appropriate levels to take the place of RYR1, because that gene is different, it's intact and not affected by the disease, or would it be structurally not be capable of doing the interactions and function?
Yeah, so
before you answer the question,
yeah.
It's a fantastic question, but can you just get everyone on the same page here, just sort of briefly describe what are called these isoforms, RYR1, 2, and 3?
Yeah, exactly. So the human genome encodes 3 different ryanodine receptors, and they're called RYR1, RYR2, RYR3, right? And RYR1, it's the skeletal muscle ryanodine receptor isoform. But actually, skeletal muscle also contains a little bit of RYR3. And it's a protein, it's a gene that's very similar. If I was to look at the structure and you look at it from a distance, say, well, that looks pretty much the same. But of course, some details are different. And so to answer Dr. Bonneman's question, so first of all, RYR3 we know cannot couple with the dihydropyridine receptor. That's one thing. You could engineer it to do that, but then you might as well engineer RYR1, so it defeats the purpose. Another problem with RYR3 is that it's a protein that by itself really likes to open. So even if it was to be able to interact with the DHPR, it probably would open spontaneously a little too often, because this is a channel with what we call a very high open probability, and that's probably also not something you would want. So unfortunately, I don't think that's an option either. Yeah.
not to geek out too much on ryanodine receptors, but we have a mouse model with reduced RYR1 expression, and we tested this idea by overexpressing RYR3 in those cells. And what Philip said is exactly what happened. It was actually detrimental because it created more leak and created more problems. And it didn't enhance EC coupling. It didn't enhance the DHPR's ability to release calcium that was in the store. So it actually was not a good intervention.
Can I just make the point for those who are new to this and who have never attended a meeting before or taken a class in genetics or biology, that if you are confused, overwhelmed, or freaked out right now, that's totally normal. I've been to like 4 or 5 of these meetings. I'm on the board, and today I learned so much stuff that I haven't understood for 10 years. So every time you come to a meeting like this, or you read the patient handbook, or you read an article, or you talk to another family member, you will slowly get it more and more and more and more. And I just want to thank Bob and Philip, because today I understood my body in a way I've never understood it before. And when my daughter was young, my father used to sort of bitch at me about like getting her into more physical therapy, as if that would really change her. And I think the more you understand how your body works, and the more you sort of get it, the more you can forgive yourself for the things you can and can't do. And even if these talks don't necessarily tell you how to change your life to make your life better today, whether you should exercise or shouldn't, it will hopefully give you hope that they are working on a drug treatment or therapy, and that if you understand how the researchers approach this, you can maybe have more hope than you did at the beginning, because that's what it's done for me. And I hope that that makes sense. So don't give up on yourself if this is hard to understand. It will get easier. Thanks.
What we're talking about here is extremely sophisticated science, and so this is very cutting edge stuff. So yes, if you are confused, that's normal and probably appropriate. I'm confused. There are a lot of things that we're trying to work through here, but I would encourage you during the break or at any time, go up to any of these individuals. They're really here for you, and I think they really want to interact with families and individuals. So do not be shy. As I tell people all the time, the only stupid question is the one that is not asked. So don't hesitate to ask questions in this forum, or if you're too shy, one-on-one. But I guarantee you, if you have a question, there are probably 5 other people who have that same question. So Matt, go ahead.
Super cool stuff. Are people working, or are you working on sort of understanding the structure of ryanodine with dihydropyridine receptor, and sort of figuring out, are there interactions that people can sort of target between those 2 receptors?
Yeah, so that is essentially where the field here is going towards, because this is a big missing link, is to really indeed get big structures of the dihydropyridine receptor and the ryanodine receptor all together. And it's biochemically extremely challenging. It's not that nobody's interested, because it's a very important question. It's a different kind of challenge, and there's a new kind of methodology called cryo-electron tomography that's kind of in the pipeline that will probably resolve this question. It will probably take a few more years before there's something at high resolution where we can understand it, but definitely, because we know that the presence of this dihydropyridine receptor affects the way that RYR1 can open and close, and vice versa. So we really should see it as one big super complex, and that is an important missing link, and that will definitely tell us a lot more about disease mechanisms. Yeah.
My question is, going the medical route with medicine, but have you ever looked at nutritional intake or food and fluids as something that could maybe help support the decrease or increase of what's needed for this gene?
Yeah, I haven't. I don't know if others want to comment on that, on nutritional supplements and things like that, right? So.
Yeah, it's a great question. Do you want to talk maybe a little bit about antioxidants and acetylcysteine and Yeah.
So the one thing to think about is antioxidants, right? Because so what we know about RYR1, and this is combined work from many people in the field, is that when RYR1 is actually also sensitive to what we call reactive oxygen, right? So when your cell is in some kind of an oxidative stress, there are little modifications that can happen on RYR1. And what that actually does, it makes RYR1 open easier. So you can imagine if you already have a sequence variant that makes it open easier, and now on top of that, you have like these oxidant species that we call them, they modify it, it makes it open even better. That means you're going to have more severe disease symptoms. So there's this general notion that if you can downregulate that with some kind of antioxidants, so that was there was a trial on N-acetylcysteine, to help mitigate that. The results were not really overwhelming, right? So, but that's one general strategy is maybe think about supplements that are generally antioxidants, but there's no clinical trial evidence that it actually will work. But on the other hand, it probably doesn't hurt unless you overdo it, right? So antioxidants might be something to think about, but there's no clinical evidence, but biochemically it would make sense that it should have benefit. And it has worked in experimental models. It has worked in animals, just in... Exactly, yeah. And zebrafish, and yeah.
N-acetylcysteine, NAC, you can go to any supplement store and buy it. N-acetylcysteine, NAC, or NAC. You could Google it and buy it. You could probably get it on Amazon right now. Now, there's a certain formulation, and I'd have to look at the details about how it's that you want to get that was used in the clinical trial. I'm not sure there's anyone from that clinical trial. Was Irene here? But anyway, if you want to email me, we can get you that information. Question?
Hi. So I was looking at the structure on the 3D model, and I was curious, surrounding the center, I guess you could say on the arms, there was these 4 holes. What is their purpose exactly?
Yeah, okay, you've got a good eye, right? So there is only one hole that goes through the membrane, right? So the membrane that separates the sarcoplasmic reticulum and the cytosol, right? There's only one hole that goes, that goes through there, and that's the hole that opens and closes, right? But indeed, if you look a little further, you see kind of this giant openings, right? It's not like one massive block. It's very, I guess we're not really certain what it means. It does mean that the calcium that comes out, right, as it goes, it doesn't have to go in a straight path. It can kind of, go a little bit, escape in various ways. And it's sometimes called like the shower head model, right? It's like it can escape in multiple ways. But probably what it also is doing, by having holes, that also means you've got a lot more surface area. So when you have like a big block, right, you can only bind things on the outside. But if I make some holes in there, you have additional surface where molecules can bind. And we know that in some of those holes, they provide access for other molecules that are regulating the function of RYR1. And in some cases, we know which molecules those are. In other cases, we're still trying to figure that out. But that's probably the reason, is to maximize the amount of surface to which molecules can bind and regulate how RYR1 functions.
Awesome. That's so cool.
Philip, thank you so much. Dr. Bonpetigan.